So what are zymogens? We learned that the zymogens are the inactive form of enzymes. There are many enzymes which are formed in their inactive form. So the inactive forms are called as the enzyme precursors also, and they are converted into active enzymes in the presence of a specific factor. It can be a specific pH or a specific substrate or a specific enzyme activator, or it can be enzyme, another enzyme. Enzyme precursors are generally longer than so they are bigger in their size or longer in their size compared to the active enzymes, and then they are made up of protein. For example, pepsinogen changes into pepsin, inactive form, active form, or you can say zymogen and the actual enzyme in the presence of HCl activated. Similarly, trypsinogen changes into trypsin, chymotrypsinogen into trypsin. So these are the inactive forms and these are the active forms activated in the presence of certain factors. It can be pH or enzyme or otherwise the substrate. So remember, this conversion with the help of trypsin is called as the autocatalytic activity. Because trypsin has this ability, once a small amount of trypsin is formed from trypsinogen, it can start its own auto activation. Now the next topic to start is the kinetics of the reaction. So remember, this concept is in your chemistry, in the chemical reaction speed and all. This whole concept would be in greater details. I will be covering only the biological portion of this topic. So the kinetics of the reaction. Now this graph represents that this is the direction of the progress of reaction and this is the potential energy. So we all know that the molecules around us, they carry certain amount of energy. So for all the reactions to occur, the collision is required. So collision requires the energy. After picking up the energy, the reactants or the substrate, they react to form reacts a much higher energy state, which is called as the transition stage. So this level is called as the transition level. Transitional energy level or state. And the chemicals react only in their transition state. So reaction would occur at this stage only. It is called as the kinetic or the collision theory of the reaction. Energy difference between the non-reactive and the reacting chemical is called as the activation energy. Now this is non-reactive state, this is the reactive state. So the difference between two is called as the activation energy. It is required by all the chemicals, whether reaction is exothermic or whether the reaction is endothermic. Some reaction, whether the reaction is spontaneous or exothermic type, or whether it is the energy requiring endothermic reaction, all reaction needs it has a certain difference between the actual state and the transition state. So that difference is called as activation energy, and that much energy is required, then only reaction can be started. The inability of the reactant to undergo chemical change due to the requirement of the energy is called as the energy barrier. So if this barrier is not broken, reaction will not take place. So activation energy is required. This energy is required to break this energy barrier. Now the main point, that activation energy and enzymes. So what is the role of enzyme here? Now we know there is a great difference between the energy of the substrate level and the transition state. So what exactly enzyme do is, once enzyme come into picture, the amount of energy required, that means the activation energy level comes down, or you can say transition state can easily be acquired by a supply of a smaller amount of energy. So that means the energy requirement or the barrier, which was a bigger barrier, that barrier becomes reduced in the presence of the enzyme. So activation energy is the energy required to overcome the energy barrier of the reactants and make them reactive to start a chemical reaction. And energy, this activation energy is generally quite high. But what happens is, The enzyme lowers the requirement of this activation energy. So this is the reason why enzyme play an important role and faster up the speed of the reaction. An enzyme mediated lowering of this activation energy can easily be seen in this picture. Now they need a lesser amount of energy. This much energy is required now, and then reaction will occur and finally the product would be formed. I hope this diagram is clear to you. So this gives you the idea of the kinetics of the chemical reactions. Now remember, the next main topic to start is the what are the two hypotheses to explain the reaction? How does reaction takes place? So two hypotheses are there. There are two main views about the mode of the enzyme reaction, enzyme's activity. One is the lock and key hypothesis and the other is the induced fit hypothesis. Lock and key was the favorite one for a very long time. It was introduced by Fischer in 1894, but later it was found some of the properties of the reactions were not clearly explained by the lock and key hypothesis. So the new hypothesis now, which is able to explain all the functions, which is able to explain all the aspects of the biochemical reaction, is the induced fit hypothesis, which was introduced by Koshland in 1959. Remember, there are major differences between these two, which I will explain to you. So what exactly happened in the lock and key hypothesis is, according to lock and key hypothesis, long time back also I told you compatibility. Lock and key are both compatible to each other. The lock will have a certain place which has a particular shape and the levers, and the key of the similar type is supposed to enter inside, and then that's how the lock is going to unlock. So this mechanism is called lock and key hypothesis. In the same manner,